So if the structure is clear, the structure of the globule, now we will study the development. Okay, how this globule or the antheridium is developed. Now, as I had mentioned earlier also, in this case, when you see this globule, it develops on the node, okay, on the node of the uh, limited branch, right? It forms on the node of the limited branch. Now, the a peripheral adaxial cell of the node, okay, it will function as the antheridial initial cell. Okay, so the adaxial cell of the node in the limited growth, limited branches, it will act as the antheridial initial. So you can see here in this first figure here, the initial cell, okay, the figure A. So this will function as the antheridial initial. Now this antheridial initial, what happened if you take a look at figure B, it will divide transversely into a basal pedicle cell and a terminal antheridial mortar cell. Okay, so it will divide transversely into a basal pedicle cell and a terminal antheridial mortar cell. And this pedicle cell, it will not divide further. Okay, it does not divide further and it will form a small stalk cell. In the major antheridium, this pedicle cell, it will form a small stalk cell. Now, whereas the antheridial mortar cell, okay, just take a look at the figure B and C, you will see the antheridial mortar cell. It will then divide by two vertical divisions. It will divide by, in first vertical division, it will give rise to two cells. You can see it has divided transversely, the antheridial mortar cell. And in figure D, when it further divide, it will further divide to give rise to four cells. Thus, by division of the antheridial mortar cell, okay, by two vertical divisions, it will form a quadrant. Quadrant meaning four cells. Okay, so each of these four cells again, they will divide by a transverse wall. And thus, if you see here in figure E, we have 8 number of cells are formed. Okay, in figure D, you see this quadrant, that is each of these four cells, they will divide by the formation of a transverse wall and leading to the formation of 8 cells, 8 number of cells, that is in figure E. So this stage is referred to as the octant stage. Why octant? Meaning 8. Since 8 number of cells are present now, so this stage is referred to as the octant stage. Okay, so now what happened? In figure E, you can see the antheridial mortar cell. It has divided to form 8 cells, referred to as the octant cell. Now, each cell of the octant, it will further divided periclinally. Okay, as a result, we will get two layers of 8 cells. Two layers. Why? Because each cell of this octant stage in figure E, it will further divide it periclinally to give rise to one more layer of 8 cells. So now I will show you this one in figure F here. What do we have? We have two layers of 8 cells now. Okay, so in this way, again, these layers, the cells of the inner or the outer layer, it will again divide periclinally to give rise to one more layer. So just remember, in this way, 3 radial layers of 8 cells each are formed. Okay, so just remember, in globule, in antheridium, there due to the division of the antheridial mother cell, we will finally it will finally give rise to three layers of eight cells. Okay, so there are three layers of eight cells are formed, and actually the cells of the outer layer, okay, the outermost layer, these eight cells, they will transform into shield cells. So you can see in figure G here. Okay, just take a look at figure G. Now, those eight cells, okay, the outermost layer, because we have three layers, just remember. So the outermost layer, it get transformed into shield cells. So we have eight number of shield cells. And what happened to the cells of the middle layer? The cells of the middle layer, it will elongate towards the center of the antheridium, and eventually it will form the manubrial cells. Okay, so like I had already explained in figure H, the manubrial cells. So just take a look at figure I here. You can see, since from one shield cell, this is a single shield cell. Okay, so from a single shield cell we will get one manubrial cell will form. So since we have eight number of shield cells, so there will, from each cell, there will be eventually eight rod-shaped manubrial cells, eight number. Okay, so that is the middle layer. Now coming to uh, the last layer, okay, the innermost layer. Now the innermost layer, the inner layer present at the distal end of the manubrial cells, that layer, it will give rise to the primary capitulum cells. So you had already studied here uh, in figure H the structure. Remember, from the manubrial cells we will get these primary capitulum cells, right? So these primary capitulum cells, they will form from the innermost layer. So we have the outermost layer forming the shield cells, the middle layer, it will form the manubrial cells, and the innermost layer, it will form the primary capitulum cells. So here we have three layers of cells, right? So the shield cells with many enfoldings, you can see these shield cells which are formed, they has many enfoldings. Okay, you can see here in this figure, which form various types of ornamentations on the surface of the globule. But inside, what do we have? What do we have inside? In figure I, you can see from only one shield cells, we will have the manubrial cell and the primary capitulum cell in the middle. So from the primary capitulum cell, now what happened? This primary capitulum cell, it will divide to form six secondary capitulum cells. Okay, so there will be six numbers of secondary capitulum cells. You can see here, these are the secondary capitulum cells, and they arise from the primary capitulum cells. Okay, there are six secondary capitulum cells, and sometimes these secondary capitulum cells, they may also divide again to form tertiary capitulum cells. Okay, sometimes it happens that these secondary capitulum cells, they may again divide to form tertiary capitulum cells, but in this case we have only the primary and the secondary capitulum cells. Now each of this secondary capitulum cell, like I had mentioned earlier in the structure, that they will give rise to form, they will divide repeatedly to form 2 to 4 long multicellular thread-like filaments. You can see these are the thread-like filaments known as the antheridial filaments, okay, or known as the spermatogenous filaments. And each of these antheridial filaments, I had also mentioned that they have about 200 25 to 250 uninucleate cells. So just take a look at this figure J here. Okay, the figure J, you can see uninucleate. Why? Because there is a single nucleus present in each cell. So there are about 250 cells, and these cells, they are uninucleate, having single nucleus. Now in figure in figure K, now what you see here, the cells of these antheridial filament, they will act as sperm mother cells. Okay, they will function as sperm mother cells, and the protoplasm of this sperm mother cells in figure K, okay, the protoplasm of these cells, they will develop into a single spirally coiled biflagellate antherozoid. So inside each cell you can see the antheridia. Here the antherozoids will form. Now just take a look at figure L here. The antherozoid which is formed, it is spirally coiled and it is also biflagellate. Why? Because you can see here there are two numbers of flagella, bi meaning two. So it is biflagellate and it is spirally coiled. So the process of this antherozoid development is termed as spermatogenesis. Okay, so inside these sperm mother cells, the antherozoids will form, and how these antherozoids are liberated. Okay, how they are liberated to the outside environment. Now, what happened? In the liberation of antherozoids. So at maturity, what happened? The shield cells, okay, these shield cells, because now they are intact, okay, they in this globular structure you can see all the shield cells, they are intact. But what happened at maturity? These shield cells of the antheridium, they will separate from each other. Okay, just like you can see here in figure I, single shield cells. So in this way they will start separating from each other, therefore exposing the antheridial filaments to the outside environment. Now the sperm mother cells, it will gelatinize in water. Okay, and the antherozoids, they will be liberated from the antheridia. So this is how the antherozoids, they will be liberated to the outside. The shield cells will open up, will start separating from each other, and they will expose the antheridial filaments. So the antheridia, the sperm mother cells, they will uh, liberate the antherozoids inside the water. So this is the whole process of development of antheridium, that is the globule in Chara.